Welcome, this is Mr. Buss, and in this video, of course, I'm going to go over the 6A DNA and Replication notes, similar to what I would have done in class for notes, but of course we're not in class now, so hope this is helpful. Let's go ahead and get started. So DNA structure is the first part, and this is going to be like similar to what was in the iBook and videos as well too, but what's a nucleotide made up of? This is made up of phosphate. Phosphate is made up of a bunch of atoms together, phosphorus, oxygen, and hydrogen, and then it's made up of sugar. There's 5 carbons on that sugar. 1, 2, 3, 4, 5. And then it's made up of a nitrogenous base there as well. So that's going to be one of 4 structures, and it's called the nitrogenous base because of all the nitrogens. What's with numbers like 5' and 3'? Again, the carbon atoms aren't shown, but every time that there's a little bend there in that structure, there's a carbon atom. Also, like, not all the hydrogens are shown either, but they're numbered 1, 2, 3, 4, and 5, and that's important because there's directionality with DNA sides, and so we'll want to look for the 5' and 3' carbons quite often. What's a phosphodiester bond? This is a review from Unit 1. If you remember, the dehydration synthesis reactions for any biological macromolecule like carbohydrates or lipids or nucleic acids or proteins. But that's just where you're going to take an OH and an H, and I don't remember which one it is. I guess it's, yeah, looks like that. The OH and the H there are going to form H2O. So it's a dehydration synthesis reaction, and the new bond between the oxygen from the phosphate and the carbon there, right there, that new bond is a phosphodiester bond. If we went in the opposite direction, that would be a hydrolysis reaction. So, kind of a cool picture of DNA. There's a lot going on here. First of all, we'll talk about maybe the directionality. So the 5' end is called the 5' end because the 5' carbon is closest to that end of the left-hand side. And DNA directionality goes from 5 to 3. So this side, the direction is going that way. And then this side, since the 5' end is down here, there's the 5' carbon. And then, of course, the 3' carbon's right there. So on the right-hand side, the directionality is that way, and that refers to the direction that that side would have been formed. All right, and then the phosphodiester bonds, right, are kind of between the phosphates and the oxygens. I mean, and the carbons, right? Phosphodiester bond there. And between, in the middle of the DNA, you've got a lot of hydrogen bonding, and I just think of that as like a magnetic. So when the sides are close to each other, if they complement, they just snap together. And complement means like A complements T. Adenine and thymine complement each other, and guanine and cytosine complement each other. And they just kind of fit together perfectly so that you get these hydrogen bond interactions. And it has to be the right nucleotide. So like adenine and thymine complement, and it's just, you get 2 hydrogen bonds. Now that's less than guanine and cytosine. So guanine and cytosine is actually a little bit of a stronger interaction. So that does matter occasionally. So you get 3 hydrogen bonds between guanine and cytosine. And a bond is always between a purine and a pyrimidine. So the way I think about it is the smaller word goes to the larger structure. So like adenine and guanine are purines, and the pyrimidines, larger word is the smaller structure. The thymine and the cytosine are the pyrimidines. And again, anti-parallel means that they run in opposite directions. Each side runs in an opposite direction. I'm really not going to go in super detail. You could, if you're like interested in the history here, just because it's not really on the AP test and stuff like that, but there's videos out there. So if you just Google like Hershey and Chase, there's a good Khan Academy one, stuff like that. There's a lot out there. So I'm just going to gloss over these briefly. Historically, A lot of this stuff was happening in the 40s and 50s, and like one of the famous examples early on, before they knew what the hereditary material was, they did experiments like this one, which seems cruel, but they would take like 2 different strains of a virus. So like the one here, the rough strain, they injected a mouse with the virus and it lives. Good for the mouse. And then if they take the smooth strain, the mouse dies. Okay, so we have a version where the mouse will live and a version where it dies. And then they said, okay, so let's come up with some like different like ways that we can kind of mess with this to see if we can figure out what the hereditary information is. So then they said, okay, if the smooth strain right here, if it kills the mice, what if we heat it up? Because they knew that like heating proteins will denature the proteins. So they heat up the smooth strain and yay, the mouse lives. Okay, so destroyed the protein structure, but what if we mix the 2 things where the mouse lives? What if we mix the rough strain and the heat killed smooth strain? What's that all about? Well, the heat killed smooth strain would have had DNA in it, or RNA, I don't know if it was DNA or RNA. It would have had nucleic acid that wasn't necessarily destroyed, that could have gone into, transformed into some of that non-virulent rough strain, kind of making a hybrid and causing the mouse to die. And so that case, this experiment just kind of showed, look, proteins don't appear to be the hereditary material. It's probably nucleic acids. So Hershey and Chase experiment just kind of showed that, hey, nucleic acids are probably what's determining heredity and not proteins. And then Chargaff's rules were experiments done that just found that, like, in all species, there's certain amounts of adenine, guanine, thymine, and cytosine. But the interesting thing that Chargaff found out was that if you have, like I'll just say, if you have like 20% adenine, you're going to see 20% thymine as well in that organism. Okay, and that would mean that you'd have, 20 and 20 is 40, 60% left, and the rest would be divvied up evenly between guanine and cytosine. So I just wanted to kind of point that out. That was kind of just a way to determine the base pairing, G to C and A to T. Rosalind Franklin did a lot of studies where she took crystallized DNA. So there's a process to take the DNA and crystallize it, and then it's called X-ray crystallography is the technique, and then you shine a light through that crystal, and then the light diffraction pattern, which obviously is in the picture there. I can't read that. I don't know how to read that. You have to be a specialized technician, and she was an expert at it, and she was able to look at that, and because she was really good at X-ray crystallography, she was able to determine that the structure must be helical, have these certain dimensions to it. So we're pretty close to knowing the structure of DNA at that point, and then Watson and Crick, who seem to be the most famous of the bunch, they wrote an article that determined the structure. They did a little model building, right? So they said, okay, A to T, G to C, we know there's phosphates, sugars, and they basically worked on all the angles to see if it would fit together. And in that 3-dimensional model building, which was not really done at the time, that really was helpful to them to be able to kind of show how DNA could be structurally oriented, and also the fact that it looks like it's conservative, right? If you rip the structure apart, it kind of showed how you could theoretically build the other side up. So it was kind of good at showing that as well. So anyway, just moving on past the history part. So DNA replication. So we have DNA in our nucleus. We're eukaryotic organisms. We have cells with nucleus. We have a lot of other cell organelles. Why would our DNA ever need to replicate? Well, if you ever need more cells. Why would you need more cells? To replace damaged cells, for a young person or organism to grow up and develop into an adult, to get larger. So we need more cells to replace damaged cells and old cells, and not all our cells do DNA replication, right? Some of them enter a state like in our liver or nerve cells where they don't grow and they don't divide. But in DNA replication, again, the term semi-conservative. First, the fact that, I'll use opposite directions here. When it opens up, each side will then form a new strand. So each new strand has one old and one new. So here you have like old strand, and here new strand being formed in the 5' to 3' direction. And here you have the old strand, and here you have the new strand being formed in the 5' to 3' direction. So semi-conservative because you get one of each. Old, new, old, new. And now this picture really shows a lot. So if you can look at this picture and talk about all of these, talk them all out, then I think you're pretty good at this stuff. So I kind of put bold on all this stuff. I'll just kind of go through this picture a little bit. It's a major enzyme would be helicase, right? And helicase does the unzipping. It kind of rips across the DNA and breaks the hydrogen bonds, which they're weak. It's a weak interaction, but they're significant because there's so many of them. They add up. But it's like I think of it as magnetic. As it gets closer, it snaps together, and then it unzips it and pulls it apart. What causes it from snapping back together again? These single-stranded binding proteins. So they just hop onto those single strands, and then they prevent the reattachment after the helicase does the unzipping of the DNA. And once it's unzipped, then you need some primase and primers to be added. So those sequences on the DNA are going to be looked at, and so like this would be a primase enzyme, so it's going to hop on and it's going to add a little RNA primer there. And it's kind of weird that well, wait a minute, why RNA? We're talking about DNA. That's just the way it works. There's little teeny sections of primers that are RNA, and they'll be later removed when it's all said and done, but they are needed to get going because DNA polymerase, which is shown on this side here, DNA polymerase, I'll just write DNA pol, and it's DNA polymerase 3. DNA polymerase 3 is going to need to start somewhere. It has to hop onto a double-stranded section, so you need those RNA primers to get it started. And then, like the one that I just was looking at here, the DNA polymerase here is moving in this direction. It's making a new strand in the 5' to 3' direction, and it's going towards the replication fork. So it's just going to keep going. As long as that helicase keeps going, this is the leading strand. It's going to keep leading towards that replication fork, and it's continuous. It's going to keep going all the way. So that's only going to need one primer, one primase enzyme, one primer, and go. All right, and then this top section of it is going to have the opposite problem because DNA is anti-parallel. So this one's going to have to constantly hop on and then kind of move over and look little sections and chunks. So here's DNA polymerase 1. All right, and then DNA polymerases. Yeah, what else am I missing on here? Oh yeah, ligase is gonna. Here's a ligase here. When there's a gap and a phosphodiester bond needs to be fixed, like if you remove a primer and need to make a connection between, like here, it needs a little a connection there and stuff like that. So the ligase is going to form that phosphodiester bond between nucleotides. This is kind of a cool feature here. If you overwind the DNA, which would happen because as the helicase is moving across and it's pushing on, it's twisting and unzipping, but it's also forcing a supercoiling. It's forcing this section of it to wind tighter, and if that happens, it's going to start to form all sorts of weird loops and stuff like that. If you've ever done that and twist something a lot, it's going to twist it too much. It's going to start to form these weird loop interactions, and so to prevent that from happening, and it'll also slow the whole process down, Topo Isomerase, which is this right here, it does a real cool thing. As it gets over twisted, it makes a cut on one or both of the sides of the DNA and doesn't let go. It cuts and then lets it unwind, unravel a little bit, and then it reglues those, and then it'll kind of keep moving along. It'll keep moving along as this process occurs, or it'll hop off and a new one will hop on once a supercoiling interaction occurs as well. So that helps, because otherwise it's going to get too coiled and wound up and it's not going to work very well. But this process happens really, really, really fast. Where do these new nucleotides come from? They just come from the food you eat. Like if you eat strawberry, you're eating cells that contain DNA, and your body will break apart that DNA into the nucleotides. They'll be floating around in your cytoplasm and in your nucleus, and they'll be utilized. So if you don't, I guess if you never eat anything that contains cells, like if you've never eaten a carrot or broccoli or strawberry or something like that, then you're going to have to possibly get your DNA from synthesis. So you'll have to synthesize your DNA nucleotides. Replication, where does it begin? It begins at a special place called the origin of replication. And so it has to have, like helicase here, has to have the unzipping have started already. So you have these complexes that like wrap onto the DNA and like pull, and they pull enough to like, it's like, I think if you ever try to blow up a balloon that's hard to blow up a little bit, you have to like stretch it out a little bit, and then, oh, there, you got it. It's like that. You kind of have to like get that initial force started, and then once it pops open, then you can have these other complexes hop on, and the single-stranded binding proteins hop on and stuff like that, and then helicase can go in, but it has to have that happen at an origin. And the origin of replication is unique because it has a bunch of AT regions. Why is that important? Oh, remember, A to T only has 2 hydrogen bonds rather than 3. So they're weaker sections. If you have a bunch of A's and T's, they're going to be at the origin because it's going to be easier for it to pop apart because there's not a bunch of G's and C's which have 3 hydrogen bonds. Just in bacteria, like you need to know, as AP Bio students, you need to know that bacterial chromosomes are circular and that they have one ORI region, and so it'll go around and it'll replicate that. We have a lot more DNA, so it would take forever to replicate our chromosomes if it just had one. So we have placed along various sections, we have multiple origins of replication, and then those bubbles grow, and then of course when they meet, then it's completed. And then here, these pictures just kind of go through the process in a little more detail than what I talked about up here, kind of just isolating them. So like shows the primer here is, remember, an RNA primer added by a DNA primase, and then the polymerase can hop on at that section, and then it can start to move across. And then polymerase can only add new nucleotides to the 3' end. So that's why it works and forms new strands in the 5' to 3' direction, because it can only add a new nucleotide at the 3' end. Yep. DNA polymerase 3, it's called. Is what does that. Then you get the leading lagging strands. Okazaki fragments are the lagging strands. All right, that looks good. And then I don't think I mentioned earlier, I wrote down DNA polymerase 1, but DNA polymerase 1 removes the initial primers and replaces them with the correct DNA nucleotide. So 2 major DNA, there's more than that, but there's 2 major ones to learn about. DNA polymerase 3 is the major one that does the huge amount of replication, and DNA polymerase 1 just is this kind of small healing at the primer where it removes the primer. Why does it have to remove it? Because it's RNA, adds the DNA nucleotides in place there. And ligase connects the Okazaki fragments, so those phosphodiester bonds between the Okazaki fragments of the lagging strand. So yeah, DNA replication, DNA and DNA replication. Hope me just talking about it was helpful. It's pretty similar to what you had in your videos from outside sources as well. So hope you're doing well. Take care, and check out the next video is RNA and proteins.